What else you have studied today? Tell me about something more. You have studied gone through the budget. Budget, what are the highlights? How was the budget like? Good budget or economic survey? I think came on day 4 yesterday. Who presents the economic survey? Who presents the economic survey generally? Ministry of Finance. Ministry of Finance economic advisor generally. Yes. Tell me.
National growth
National growth nominal which is possible. 9.2%.
GDP का बढ़ाया 8 से लेके 8.5 तक. तो जो बताया था कि एग्रीकल्चर सेक्टर 3.5 से 3.79% ग्रोथ हुई.
Nothing. It is nothing. 3.7 growth is what is this thing? Inflation is more than 5%.
पर उन्होंने बोला
You understand inflation? What is inflation?
Rise in prices.
Rise in prices. They wrote the 3.5% growth means what? Less than 5% growth. So you know.
पर उन्होंने बोला कि जैसे कोविड में बाकी सारे सेक्टर नेगेटिव में चले गए थे, पर एक ही सेक्टर था एग्रीकल्चर सेक्टर जो पॉजिटिव में चल रहा है.
What was during continuous 3.5 since now? What is the growth? 3.5 is nothing.
मैन्युफैक्चरिंग सेक्टर में अच्छी ग्रोथ है.
Okay, it's not that good, but compared to China, we are nowhere. Manufacturing, they are manufacturing hub of world.
सर्विस सेक्टर में भी दिखाई देना चाहिए.
Service sector is okay. India is doing good in service sector, but manufacturing and agriculture not good.
वो बताया उन्होंने कि CPI कंज्यूमर प्राइस इंडेक्स वो 5.5.
5.5.
और WPI होलसेल प्राइस इंडेक्स वो लगाते हो गया. डबल डिजिट हो गया.
Mainly it is due to the oils. Oils. You understand now oils? Which oils we consume? Indians consume more oils. Which oils? Generally the oils we use in our kitchen. Mustard oil, palm oil, CPO, that is known as CPO, crude palm oil. Palm oil, then your olive oil, then this, what is this? Mustard, peanut, all those different oils, palm oils and rice oil, rapeseed oil, these all oils, these constitute, I think the third largest import, second largest import category. Then import categories. After China, India imports the metals. But I am talking about the goods being imported as per the goods. So most like crude oil is the one you use in your vehicles. It is the largest import. So we import that consumes the largest amount of currency, foreign exchange. After that, I think the electronics, and the third is this palm oil. Then second is gold. Then electronics and this oils. So these oils have become very expensive. Mustard oil, I think I used to buy mustard oil for 140 rupee per liter. Now it has become 210 or 220 per liter within a year. So inflation has been anything like in edible oils. The government say we have controlled the inflation. What way you have controlled the inflation? No inflation. Prices of tomato and other also have these consumer commodities have increased. The people who like you are using this product, the price rise is like anything. The prices have risen anything, and the government has not given any relief in any form. So budget, you tell me what budget it is? Nothing. No mixed budget. It is a kind of mixed budget. No growth. They put a lot of money in infrastructure. Parvatmala and this that. But what about
जितना हम कैपिटल पे इन्वेस्ट करेंगे उतनी एम्प्लॉयमेंट करेंगे.
From where the employment will come? Employment will never come through this government sponsored government spending. Government spending creates demand, but main demand will come from the private sector. Private sector is not doing. We are not spending money.
So
PPP model is okay. That is for certain things. You for road, proportion, you can. But generally, in like manufacturing, big chemical factory will be set up. Only private organized can only invest that money. Government should not investing in those things. But that investment is not coming. Private sector is tight fisted right now. Even the foreigners, they are not. What policies they are PLI scheme they have announced. PLI this production linked incentives. So what they like China this way, you won't get the supply lines of China. You have to like very innovative, but nothing innovative, no much innovation. It's a continuation of the budgets. Similarly, continuity is same every year, same type of budget. Nothing new, something new, very broad, bold. Something is not coming up. So what budget they stand up for 1.5 hours in Parliament and give speech for this much for this sector, this much for this sector. Healthcare, no, nothing much has improved. Healthcare, what improvement is there? Healthcare is in shambles.
Telemedicine.
What telemedicine? I have tried telemedicine, but it doesn't work. Sanjeevani, you might have heard about. Sanjeevani is this scheme launched by this central government only for teleconsultation. It doesn't work. You have to give your name, then you are given a coupon, then you wait for so many hours, then still nothing happens. It's not instantaneous. You like you go talking, picking up phone and ringing and somebody asks you. Just like schemes. Practically, healthcare, what is there? In J&K, we don't have a private sector. No Apollo is there, no Fortis is there, no private hospital is there. Only government medical college and Narayana is there. So what interest, like, where the people are? So people who are willing to spend money, even they are not getting good facilities. And this Narayana charges like anything. So where the people go? And medical college like a cattle shop, cattle shed. So what kind of, I don't know what planning they are doing and what money they are spending. Money is not a problem, but planning is totally poor. They are not able to, now they are trying to have this AIMS at Vijaypur. What, it is taking so much time. I'm just flabbergasted. What governments means people they should be accountable like people who are doing all this. But things happen, no, but only Vikas push. What Vikas? What Vikas is that? These are so simple things. These are not very complex things that they will take a lot of time and money. These are very simple things. If you put your will to do something, it happens. But they are not doing. I don't know. What is the meaning of budget? Budget, I told you that it is budget. It is a word from French. Budget means leather case. Leather case which this, no, she is carrying Bahi Khata, that format. But leather case where the papers of this proposal for the next year would be posted. Budget means the income and expenditure statement of the government. Now this is the income government is estimating that this much. Generally there are three types of accounts. There are original estimates, then budgeted estimates, and then revised estimates. Means that how much you have planned original was this was the original, then actually what has happened, how much you have spent, that is the revised estimate. So budgeted, so that those estimates based on those they plan for the next year. This much is needed for this sector, this much is needed for this sector. So this expenditure they plan based on the estimate. So estimates the receipts you get from the income tax direct and indirect. Expenditure is the money you spend. Defense you spend and so many social sector and infrastructure. So these are the different things. Budget you must know. Like budget, how, what is the procedure? Generally it is presented in the parliament. Now she has made a speech. This is a budget speech. After that, there will be vote on account. Not vote on account. Demand for grants will be presented by every department that this much money we need. On that discussions will be had and then passed, and then parliament passes the demand for grants, and then they get the money authorization to spend the money. So this is what budget, legislative processes of budget. This is all English tradition. So we have all got it all from the Britishers. Earlier, what Indian used to be there, like there used to be a court of the king budget. There won't be any budget, like only whatever would come would be spent as per the will of the king. Now the legislative processes have settled. Now the parliament is the hold the purse strings. Hold means it authorizes expenditure, the money. Otherwise, not a single paisa can be spent without the approval of the parliament or the state assembly. So these are important things you should budget. I won't ask you about much about in prelims, mains they may ask. Then this budget they may ask capital budget. What is revenue budget? What is capital budget? What is zero based budgeting? What is performance budgeting? These type of things they may ask in questions. Anyhow, so anything to whatever I have lectured on biotechnology, any doubts? You have any doubts? Something you want to know? Anything you want to know? Biotechnology. You followed that my lectures on this this videos? No, not a single. No problem. You? So then you missed a lot of lectures, no? Biotechnology, I think. What? Signal is a problem. Yes. Okay. No. I told a lot of things about biotechnology from some other places. I got material from some other places. No, I am proceeding according to what is in your notes. Notes you have got from this institute. So I am telling according to that, so that you become comfortable. That's what I am trying to do. You become comfortable because you are comfortable with the notes. You have the notes. Sorry. So you know what to read. So that way I tell you, because I told a lot of things about biotechnology which are not in these notes. These are just simple notes, very simple notes, like they are just collected some data information from some places and organize them into this one big note. So I'll go according to those notes, because I told a lot of things about biotechnology, what this is all about. They have simply written the definition that this is a set of techniques involving manipulation of genes and all those things. So I told you it involves a lot of things. It involves immunology, protein engineering, then plant hybridization. So a lot of things are there. So these are very simple notes. So I try to give additional information, but I go according, I've covered that part, I go according to the notes. But if you have any questions, do ask me, because biotechnology is important. This year the question was there from biotechnology in GS. So I told about the biotechnology in the Indian context, like how biotechnology is being used in the Indian context. That is DBT, Department of Biotechnology, has lot of projects in mission mode. Oil palm, we talked about the edible oils. Now oil palm, there is a oil palm project, National Edible Mission. What is that? So this is also where biotechnology is being used. India is trying to know, like I told you about the defense, import substitution, indigenization of technology. Like they try to develop the technology locally, because technology is expensive, importing is expensive. So like Rafale, they bought it for I think 70,000 crore rupees. So that is a lot of money you pay. So instead, if you manufacture locally, you will be paying the local companies. So you will be saving a lot of money. So same for this to decrease the cost of import of palm oil. So they are trying to have a lot of areas covered under the oil palms, cultivation of oil palms, particularly among the coastal areas where the oil palms are grown. So they have a Department of Biotechnology, through bioengineering, through biotechnology, they are having best this palm plants. So they are covering those areas. So National Edible Oil or National Oil Palm Mission, this is a particular project being run by the Agriculture Ministry in association with the DBT for production, for increasing the production of oil palm in the country, so that import substitution takes place. So these are some missions you should know. National Oil Seeds Mission. Do go find out from the net. National Edible Oil Mission. Edible oils include mustard, like in Punjab and other places, a lot of mustard is grown. So that to improve the varieties through biotechnology. So you improving the plant varieties, increasing the yield, making the pest resistance, drought resistant. So those kind of things. Herbicide, less herbicides being used, herbicide resistant. So that's what biotechnology is helping in this mission. So Department of Biotechnology takes a projects. I told about sericulture, aquaculture in biotechnology. So these, like they improved fish, they would put improved prawn, sericulture, improved this silkworm, they would be using, that increase the yield through biotechnology. So these are some projects. That is the applied aspects of biotechnology, which question you got in this mains this year. So I'll just repeat this thing. This is very generally thing. generic thing. I don't know, this helps you because you have to develop the concepts. If you develop the concept, then you will be able to answer GS questions very easily. But still, this information is sufficient, I think, is relevant because you have the notes, you are going through notes only. You tell me, what do you understand by biotechnology? I told us certain things. You have written the definitions as well. So what do you understand by biotechnology? Tell me, so I know where you stand. I told these were I gave you the definition and this is as per your notes. I gave 3 definitions. The definition as per your notes refers to the application of various biological living organisms and processes for the production of useful substances or effects in medicine, agriculture, industry, etc. It includes various laboratory techniques which enhance the usefulness of biological organisms and products for humanity. So this is a definition in your notes. But I told a lot of things that it involves protein engineering, then cell culture, immunology, fermentation technology, genetic engineering. So collectively, all I told you, that is your biotechnology. So biotechnology is not just you are using certain techniques in agriculture, you can use in aquaculture, you can use in animal husbandry, in environment, so many other aspects you can cover this biotechnology. Still, you ask me questions like you have what perception you have of biotechnology. What do you think? Biotechnology is? Give me idea so I know what is in your mind, like how you have perceived the subject. So I clear the concepts. So I build your concepts. So you are able to answer the questions in examination hall. Tell me, what do you understand by biotechnology?
Biotechnology, it's practical knowledge, practical knowledge like practical techniques, practical experiments, practical knowledge like engines.
Engines?
I mean, engines like railways, the techniques that are used in that. It's
Biotechnology you using in the railways?
sorry, sir, it's science only.
Science?
Science technology.
No, see, bio.
Biotechnology means technologies in sir, like microorganisms. How?
Very good. Bio means that's what I am why I am asking you question, because I can go, I can be like a railway engine. I can just start and go on, and you will be left behind. You won't be just following the engine.
When technology begins.
Okay, no problem. That is also technology. Technology is what? Technology is a practical techniques used. Bio means living,
Living.
and technology means techniques you are using. So your techniques you are using on living things or through living things. So bio, I told you about that we engineer bioengineering. We have engineering bacteria. Like engineering, I am constructing this building. Engineering, I am making a fan. That is engineering, electrical engineering. So bioengineering means I am manipulating the living things, microorganism I am talking about. So I am manipulating the living bacteria, viruses. So this is biotechnology. Like this virus is coronavirus. This is also biotechnology. This medicine they have made in response to the corona pandemic. This is biotechnology. So that's, you tell me what biotechnology.
So biotechnology is the technology that we use to modify or make new organisms
Mm-hmm.
or microorganisms,
Mm-hmm.
or to modify the plant structures.
Mm-hmm. Plant structures or plants, good. These are all okay, living beings. We are modifying these things. And what are the techniques we use in biotechnology?
Gene editing,
Gene editing, good, very good. RDT.
CRISPR 9
Cas90, that is a gene editing only. The CRISPR is the method of gene editing. That I will tell you later. You tell me what biotechnology is?
Sir, biotechnology is using the modifications of human.
Human?
मतलब यही ये मतलब DNA modifications.
DNA?
Yes sir.
DNA what?
So modifications
Of DNA of what? Of microorganisms. Human is also that you modify through microorganism only. Okay, that is. So you should be clear about this biotechnology. Okay, tell me about the applications. I told you about the applications.
So applications, biotechnology application, green application is for agriculture, white for I think red for medicine, white for industries, and blue for marine or aquatic. Aquatic or marine are very rare. So another.
Bus. What were bus? This is bus. What you for you bus? I don't bus. I bullet train. I am in bullet train. Tell me more. So these are all the branches. These are application. I told you environment, medicine, industry, health, agriculture, aquaculture, marines. Yeah, those are the things application. Then this bus. You won't be able to answer questions like this in mains. Competitive, I told you, a lot of people from Delhi, Bombay are appearing for this exam. So if this is the way the thing, you are building knowledge, then it's no. Anyway. So tell me more about biotechnology. I told you about the RDA, Recombinant Dna Technology. What is RDA? RDT. Recombinant DNA Genetic Technology. Okay, forget it, because that's why I ask me a lot of questions. That way your doubts will be cleared. Otherwise you will be stuck with whatever is in your mind and just reading notes, and you won't be getting anything. How to organize that information in an answer form. So now I'll talk about as per your notes only. Have I talked about the tools used in biotechnology? Tools, this is as per your notes you got from the institute. So I am enzymes, yes, RATA, yes, good mugging. Vectors, competent hosts. What enzymes are?
Enzymes are basically
Enzymes are not microorganisms.
Sir, these are used to produce useful materials like insulin from.
Okay, why? Enzymes are basically body produces enzymes. They are chemicals in our body. They are sort of proteins. So these are helpful in like catalyst. You have heard about in catalyst in chemical reaction, na? That speeds up the reaction. So these enzymes are sort of those chemicals in our body which speeds up the reaction. Because our body is a chemical factory, lot of reactions are taking place, chemical reaction. Food is getting converted into energy, oxygen is getting supplied to the cells in the body. So lot of chemical reactions are taking place in our body. So these enzymes help in those in activating. Yes, these are artificial enzymes, artificial things created through human efforts. So they are also used for these detergents they can make. So I will go according to these notes. You notes are there with you, but still you write because it will help you in memorizing, because this is a memory test. The way I have understood, this is a memory test, not much of knowledge of concepts, though concepts are needed, but generally people are just verbatim writing whatever they learn by rote. So enzymes, tools used in biotechnology. This is as per your notes. If you don't feel comfortable, tell me. This is note, I'll just skip this and go fast. So first one is enzymes. Enzymes are biological substances made of different kinds of proteins. Proteins, you know what are proteins? I talked about protein engineering. So our body is what? Like when we, protein is 40%, whatever we have in edible form is 40% is proteins. You have an apple, or you have a roti, or you have rice. So these all things are made up of small biomolecules. Biomolecules. So these biomolecules are mainly of four types: proteins, carbohydrates, fats, and nucleic acids. These biomolecules, our body, these are the basic building blocks of all life forms. Nucleic acid means DNA, RNA. So these are the what we eat. When we are eating an apple, we are eating a lot of some carbohydrates, sugars. Those carbohydrates means sugars. Sugar mean not that processed white sugar which you consume. That is also sugar. So these are technically they are known as sugars or carbohydrates. We are eating carbohydrates, proteins. So these all go into our cells, individual cells. They form energy, different kind of, they form ATP energy, that is known as ATP energy, from which we get, that's how I speak, the energy I get to, from this energy I am able to speak or move or see. So this is all the processes taking place, and these are the biomolecules: proteins, carbohydrates, nucleic acids, and fats. So these are things. Then enzymes. Enzymes are biological substances made of different kinds of proteins. So proteins, these proteins are there. There are different kinds of proteins. Thousands of proteins are there in our body. Different kinds of proteins. They are the major building block of our body. Muscles have protein, and then every skin, everything has got proteins in it. So these are the structure. Cells are small things adding up to, they make up tissue, skin tissue. Small cells adding up, making up the big things. So they all have these cellular biomolecule, proteins, carbohydrates, fats, nucleic acids, and all those things. Each cell has nucleic acid, that is DNA or RNA in it. So enzymes are biological substances made of different kinds of proteins and play a vital role in various processes such as cell metabolism. Or enzymes are biological substances made of different kinds of proteins and play a vital role in various processes such as cell metabolism. Cell metabolism, you understand? What is metabolism? Cell metabolism. Cell metabolism means it's various reactions taking place within a cell. Billions of reactions take place in our cells, individual cell. Proteins are made, nucleic acids are changed, and all those things. These are too much technical, no need. So you write this, such as cell metabolism, and it increases the rate of reaction between biomolecules. हेलो, हाँ बोलो. वो नेगेटिव. हाँ 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 हाँ. कल मैंने किया था फ़ोन, फिर क्या हुआ था? काट दिया था आपने शाम को, रात को किया था ना मेरे ख्याल में. पता नहीं वो नेटवर्क, मैंने फिर किया तो नेटवर्क की प्रॉब्लम आ रही थी. अच्छा, चलो ठीक. नहीं अभी नहीं हुआ, मैं अभी क्लास में हूँ. अभी करा लूँगा, अभी जल्दी करा रहे हैं ना. मैं नहीं कर रहा ना, उन्होंने बाहर पोल पे कुछ खोद रहे हैं नीचे से, तो उससे तारें कट गई हैं. मैं क्लास में हूँ इस वक्त, यहीं पे जहाँ पे. हाँ करवा देते हैं, मुझे जल्दी हो जाए. ये नेटवर्क है ना, मैं अभी गांधीनगर में हूँ, वहाँ पे टावर नहीं होंगे ठीक. हाँ तो यहाँ नेटवर्क अच्छा है ना, तभी तो मैं उस दिन कह रहा था. ठीक है, चलो फिर ठीक है, मैं बाद में करूँगा बात. ठीक है, मैं करा दूँ. चलो कोशिश करूँगा, उनसे पूछूँगा कब तक करेंगे. ठीक है, मेरे करने से थोड़ा होगा, वो उन्होंने करना है. हाँ ना, ठीक है. I'm sorry. So you have written these are biological substances made of different kinds of proteins and play a vital role in various processes such as cell metabolism, and it increases the rate of reaction between biomolecules. So these are the biomolecules I told you, their reactions are taking place, proteins or enzymes are getting into the protein. Basically these are tools used in biotechnology. Tools means what? So like you can use an enzyme to cut DNA of a bacteria or of anything using enzymes. So these are the tools, so that they act as scissors to cutting the DNA. DNA I told you, like a helical structure, this like this. So you can join through laddering. So you can cut through these enzymes and all those. So these are the tools. So they are given the definition. Still you write, no problem. Enzymes are also used as a catalyst. Enzymes are also used as a catalyst for food preservation. Enzymes are also used as a catalyst for food preservation, making of detergents and textiles, Then enzymes are used to make cultures and are also used in the development of medicines. Enzymes are used to make cultures and are used in the development of medicine. What are cultures? Cell cultures. You have seen cell culture means like when you go for a test, diagnostic test, generally you go for a diagnostic, they take out the blood or urine or saliva or from nose swab. These are the body fluids they check for. So cultures are what? You isolate the cells and harvest them. That is, increase them. You have seen in a round dish they have certain things. They are growing the cells, cultures. So you get me? Cultures you understand? No, you don't understand. Cultures means these are like getting cells from my body. How do you get cell? You can't get a cell directly from my body. You will take or you cut a piece from my skin. When you cut a piece from skin, then you put certain things like enzymes and all those things to make those cells removed from those tissues. There are a lot of other things behind suppose this is a small cell, like this brick is there, then there is cement, then there is sand, then gravel, then again cement. So one brick is hidden inside. So then another brick, so whole wall is there. So this structure is built out of cells, skin cells, small cells. So how do you get this cell? I have to break the wall to get the brick. That is a cell. So these are the enzymes and all those things are used to get those cells, individual cells. Because from the cells we can do the engineering. Cell engineering means genetic engineering. In the cell, in the nucleus, DNA is there. So we can modify the DNA. That DNA expresses the whole form, what color of eyes we get, or color of hair or skin, or what kind of other things, traits we get. So these are in the cell, within the. How do we get the cells? Like how do we get brick? Cell is like a brick. How to get the brick? I have to break the wall. To break the wall, in case of these living things, cells, how do I break the wall? I use chemicals like enzymes to break those things, to get individual cells. When I get a lot of cells in a dish, I get a cell culture. What? Cell culture. So then I can do analysis. So this is how diagnostics has helped. You get so many reports now, liver function test, kidney function test, then so many other, complete blood count. What does that mean? They are through machines or chemicals, like in case of coronavirus. How does somebody gets corona? How do you say somebody is positive? How do you say? RT-PCR. What does happen in RT-PCR? What is the full form of RT-PCR? That is RAT. It is reverse transcript polymerase chain reaction. It is also another this kind of. This is generally what happens. Earlier what used to happen, they would put something in microscope, under microscope. So they will find whether the bacteria or viruses are there. Now the tests have become so advanced, they sort of through chemical reactions, they increase the culture, increase the cell level, increase the virus level or the say this bacteria. So you can find out immediately. If machines can find out, sensors. Not you don't have to find out through a microscope. Earlier used to microscope, microbiology. They would put microscope and see whether some virus is there or this bacteria is there. No, things have advanced. So this is because of biotechnology. So this cell culture is what cell culture is. Understood? Brick in the wall. To get to the brick, we want bricks, lot of bricks in the wall, but in the wall. In the cells in the body, how to get the cells? Break the tissues through chemicals, enzymes and all those things. Then how do you go into the cell, into the nucleus? You are using enzymes, carriers. How do you, I told you now you are editing the genes of the organizations or plants or whatever. How do you do that? How do you reach in the nucleus? Through these proteins, through these enzymes. They go deep inside. So not much, you don't need that much technology. Then comes vectors. Second part. These are the tools used in biotechnology. What are vectors? Vectors usually are DNA molecule. Vectors usually are DNA molecule used as a vehicle to carry foreign genetic material into another cell artificially. To carry foreign genetic material into another cell artificially. What does it mean? I told you, vector is what? Yes, I told you about the malaria. Malaria is what? Malaria is a disease we get. How do we get that malaria disease? Through the bite of a mosquito. What is mosquito? What is happening? Actually, mosquito is carrying a germ. I don't know. I think it is not a. It is not a bacteria or virus. It is something else. It is known as plasmid or something. It is known. So that germ it is carried by whom? That is in the blood of the mosquito. When mosquito bites me, it transfers that germs in my bloodstream. You get me? So I get the malaria. That activates and what is mosquito is? It is a vector. Vector means what? It is carrying the germ, disease carrying germ. So it transfers me, it is a vector. No, that is the method. Like when I go for a gene editing, I have to edit the gene within the nucleus of a cell. How do I do that? Through certain tools. Those tools are known as through certain carriers. Those carriers are known as vectors. So these can be anything, RNA, molecule, enzymes, and those things. Okay, vectors. Then third is the competent host. This is as per your notes. Competent host. It harbors foreign molecules. For example, a cell being host to a virus. Healthy cells need, you want to write? Okay. Healthy cells need to be made competent so that they can take up DNA. They are made competent by treating them with this specific concentration of divalent cation, such as calcium as it increases efficiency of which DNA can enter the bacteria through the pores of the cell wall. These are not that important. Host means what? Like host, somebody who is getting the treatment. So, if I am trying to modify the gene of something, say a sheep, the host is a sheep. So I have to make like somebody, you talked about organ transfer of heart of a pig was transferred, genetically modified pig was transferred to a human being. So host, who is the host? Human being. You have to make them competent, that it accepts the foreign heart, that it doesn't reject. Generally our immune system will try to reject something foreign. Say if virus or a bacteria goes into our body, immediately immune system detects something foreign has come into our system, so it tries to reject that. So but you are putting a foreign pig's heart into a human being's heart, so immune system will try to reject this heart, but you make it competent host, that you accept, no, this is not a bad thing for you, this is something good for you. So you are making competent hosts. You get me? So competent hosts, they make through certain solution these chemicals, this divalent cation such as calcium, that is, they are using tools. How cell, when there is a cell, there is nucleus, then there is a cell wall. Like these cell bricks are there, na? They have their boundaries. Na? So every boundary you have to go inside that brick, you have to break the boundary of the brick. Say you want to go inside the what inside the brick is. So you have to break it from the outside. So suppose there is a layer of cement outside the brick. That is the wall of the cell. So you have to go inside that cell. You need certain things are amenable for going into that thing. So these are what this they are talking about. This cations and all this. So you don't need to have all this thing information too much. Simple, you should understand that genetic DNA foreign, you are putting foreign DNA or a gene into something and trying to edit it. That is what genetic engineering is all about. That is what biotechnology basically is. So that those techniques you are applying in immunology, health, medicine, everywhere, your plant tissues. So those techniques you are applying there. So these are big thing I want to tell. Now you ask me any questions on this. You have not understood anything. I know that you have not understood. That's what pains me. Because if you want to clear your concern, you should ask me a lot of questions. Even silly questions you are. Tell me, ask me. Then only you will be able to build the knowledge. Ask any silly questions. How do you, like, if the question comes, what are the applications for of biotechnology for the poor people? How would you define it? These are the technical things. Sometimes they may ask you this, like blue LED, they ask this question. So they may ask you, they might ask you this question, this type of question, the technical question as well. Like in biotechnology, somebody has got a Nobel Prize, so they might ask what technique they have developed. So these things are also important, but generally the question would be like how like poor are benefited from biotechnology. So competent hosts, vectors, enzymes, you won't be able to put those things in the context of your answer for the poor people. So there you will be thinking about oil palm production, sericulture, where the employment is generated, or aquaculture, or prawn farming, oil palm missions, so those kind of things. So ask me questions. So I can, otherwise I will, it is like staring at the blank faces. You understand certain things, but do get grasp of the subject. That is more important. So you tell me how biotechnology, like what a vector is.
एक मॉलिक्यूल जो चीज़ को कैरी करता है, अगर किसी चीज़ को हमें परफॉर्म करना है, तो जैसे वो है हाउस वाइफ, तो दीज आर नोन एज वेक्टर्स ऑफ डायरिया।
Okay.
डायरिया को फैलाने में मदद करते हैं।
Good.
ऐसे अगर हमें किसी चीज़ को, जैसे हम इसमें बायोटेक्नोलॉजी में बात करते हैं कि हमें किसी चीज़ को कैप्चर करना है, जैसे सेल को हमें कैप्चर करना है, तो उस पर्टिकुलर एरिया तक कुछ एंजाइम्स पहुँचाने हैं।
Enzymes are also, in a way, you can say vectors. Means you are using vectors, maybe living organisms, to break the walls of the cell, or to not to isolate a cell, to reach that, to do some techniques, to technical things on the cell or the. Good, anyway. You tell me. You are also nodding your head. So I hope you. Otherwise, how will you build your knowledge? This is a very competitive exam. Competition is intense. Those people with biology background, they would have grasp on this subject and they would score 20 marks in this question. You will be just like doing this. Tell me. I can go very fast and finish the syllabus in one go within a month, but you will be left behind. You won't understand anything. How will you build your knowledge? Okay, any problem? That's why I'm saying ask even silly questions. Now I come to the applications of biotechnology. This is a question you had. The question in your this mains examination was, I am repeating so that you get a grasp of what I am teaching and how I am trying to connect it with your answer writing. The question in your this thing was, I have written it somewhere. So you get the basic idea. What are the research and development achievements in applied biotechnology? This is a question. what are research and development achievements in applied biotechnology? How will these achievements help to uplift the poor sections of society? So how do you link now with biotechnology? What I told you. What are the research and development achievements? So that's what I told you. You tell me. Okay, that is one aspect. Good, good. Very good. Very good. Good. Environment, industries. Okay. So this, how would this help the poor people? That when you have increased production, their incomes will increase. The employment generation will be there. So I told sericulture, oil palm, all those things increased means you have more incomes to the, giving more incomes to the, you make the medicines more cheap through biotechnology, like insulin, you are getting through bacteria, synthetic insulin. So you can decrease the costs of medicines, make it available to poor people, affordable prices. So this is a way to handle this question. Have the grasp, overall grasp of the thing, like the way you proceeded, biotechnology, this technology branches. So these are the applications. You see the application aspect. What are the research and development achievements? What are the achievements? No, the achievement they have. If you give in the general context through of the word, then Indian context, they have succeeded in splicing the gene and giving the desirable traits to the particular genes, to the particular cells. So that those cells, those modified genes or cells can be used for animals as well as for plants. In plants, in plants as well. So those with the modified plant, in case of plants, modified heat resistance, disease resistant, pest resistance, all those things, herbicide tolerant, those kind of plants, then they can increase the productivity of the farmers. In case of aquaculture or sheep, or in case of cows, they can give enhance, if you putting genes which enhance the milk production, so you are giving, increasing the yield of the cows. Sheep, wool. That is also there. You read the note, they will tell you these things. That this is a new technology. So new technology we don't know. Coronavirus, we are giving the vaccine. We have only clinical trial for 1 year had happened. So we don't know about the long-term effects of 40 years or 50 years. Only time will tell. But we are taking a chance, we are taking a risk with the technology that it is good. We are getting vaccinated, we are protected against the virus. We don't know we don't have the data of 40, 50 years. Maybe some lot of things might happen in future with this vaccination. But still we are taking a risk. So with new technology, you have to take the risk. So they might be harmful. Might be, if you have cow, they might be producing some other chemical as well in the milk. But we have to take a risk that through regulatory boards and all those things are controlled. They say, yes, it is okay for the human health. Then they say, okay. Then we take the risk and we continue living life with those risks. What way? Uh-huh. How? But you have to take your consent, na? You can't change. Okay, some dictator is there. That is illegally anything can happen. That is what regulatory I'm going to tell you, regulatory things. No, who's like nowadays hacking is happening? Who is responsible? Uh-huh. Possible? Risk is there. To go, life is a risk. You crossing the road is a risk, but still we cross the road. No, we see here and there and look here and there, and then we cross the road. Crossing, we can be hit by a road. Somebody driving rashly, they can easily knock us out. But still we take a risk. We crossing the roads every day. We do take risk. There is no regulatory body at present. That's what as per your note, there is a what is that? Genome Appraisal Committee in Ministry of Environment and Forests is there. At present, India has got no regulatory body. They have a lot of bills. They were there in the past. I'll talk about those bills. So they were passed, but at present, only for approval of this modified cotton and those things, that there is a Genome Appraisal Committee. Some committee is there in Ministry of Environment and Forests that gives approval for this kind of technology. So this DNA hacking and DNA maintenance of this bill was there, but still it has not come into force. It has not become an act. So still other countries, they have progress. They have a lot of acts and laws on these things, but India is still in the process of developing those regulatory mechanisms. You have to ask good questions. Privacy is there. Hacking, it will be there. Hacking is possible. But government will have a bill, still hacking will happen. Like I told you that day about this gender determination through ultrasound. Technology is there. People can use the technology. You can determine the gender. If it is like in some place, a female feticide, they kill the girl fetus. Why? Because they don't want a girl child. But technology is there. No? You can determine whether it is a male or a female fetus. But it is up to you. How do you know? The government has regulated it that you should not be telling the gender of the fetus. So, but that is the misuse. Misuse can happen anywhere. You can misuse the technology. Technology is there. You can misuse the technology. So, government has to regulate it. But still, people are using with the resources. They are using this. That is also hacking. They are hacking into the system to know the gender of the fetus for their own cultural things. Okay, anyway. Okay, then I come to the applications of biotechnology. I think I discussed it a lot, but I'll go through it again as per your notes, so that you feel comfortable. Applications of biotechnology. So first is application in medicine. Application in medicine. So that first part, as per your notes, is pharmacogenomics. Pharmacogenomics. That is PHARMACOGENOMIC. It is a study of how the genetic inheritance of an individual affects his or body's response to drugs. How the genetic or genetic makeup responds to the drugs. Like custom made, tailor made. You know, like when I go to a tailor, generally nowadays we buy ready-made clothes. Generally, I go to a tailor, he prepares according to my makeup, as per how my body is like. He measures all those things and then prepares the custom made. That is known as tailor made or custom made. So, these medicines, like how individually, not collectively, like 100 people respond this way to the medicine, that individual, how my body will respond to a particular medicine. So, these kind of medicine can be prepared. So, this is what pharmacogenomics is. It is a study of how the genetic inheritance of an individual affects his or her body's response to drugs. It can be used for development of tailor-made medicines, determining appropriate drug dosages, improvements in drug discovery, better vaccine, etc. This is hi-fi. Everything is hi-fi. Nothing is happening practically at the ground level. We still go for simple medicine, whatever Dolo 650 or paracetamol, whatever this crocin, we take simple, because they won't tell you, okay, you go to somebody, okay, I check you with some instrument, your body needs this thing, I prescribe this thing. You don't know, doctor only say check the pulse, say fever, Dolo 650 or crocin. So these are not tailor-made, but this is what in future, say 100 years from now, things may be like this, that you go to a doctor, he has an instrument or something, He immediately measures some biotechnological application, which immediately measures what your body is suitable to what kind of medicine. So that medicine is prescribed to you. So this is in future, but still you should have the idea. This is application. We are talking big things, but practically nothing is happening. Practically aquaculture farming is happening. Some places, small places there. So some 100, 2,000 farmers' income would increase. Not on the large scale, say 3.5% agriculture. Why, if these techniques are happening, so technology is so successful, why 3.4%? Why not 10% in agriculture growth? Because small patches things are happening and nothing much is happening on a grand scale. Okay, then part B, pharmaceutical products. This is application in medicine I am talking about of biotechnology. Pharmaceutical products. Biopharmaceuticals are large biomolecules such as proteins, and these usually target the underlying mechanism and pathways of a malady. They can deal with targets in humans that may not be accessible with traditional medicines. Modern biotechnology is often associated with the use of genetically altered microorganisms such as E. coli or yeast for the production of substances like synthetic insulins or antibiotics. It can also refer to transgenic animals or transgenic plants such as Bt cotton. So this is all those in your medicines here, like you're getting modifying the bacteria to produce insulin, synthetic insulins you are getting. So these products you are getting through the genetic engineering. So this is what biotechnology is, applications. So this is all in your notes. I'm just written that you get, these things. The biotechnology industry has also been instrumental in developing molecular diagnostic devices that can be used to define the target patient for a given biopharmaceutical. Same thing means tailor-made. Like how you diagnostic, you're taking this, some diagnostic test would be there, genetic test would be there, that would determine how your body reacts to a particular medicine. So these are the things. So the heart settings, they have given a lot of things. Then the third part is genetic testing. It involves the direct examination of the DNA molecule for the mutated sequences. Why there are certain genetic diseases? We have not, these are communicable diseases like viral diseases. Corona is a communicable disease. And non-communicable diseases are there. Heart diseases there, lung diseases, liver diseases there. So these, then there are genetic diseases. What are genetic diseases? Means you have inherited generally what happens, half of the genes we get from our mother side and half from the father side. So sometimes what happens, genes are what I told you, these are sequences, DNA sequences. So big, it is about 6 feet, is packed in micrometer. That much information is packed in a micrometer of area, more than 6 feet. Such is the length of the DNA molecule. So that what happens, sometimes some genes are not in sequence or something which is natural. So they are misplaced. So that misplacement causes some genetic diseases. Means when the genes expresses this much. Genes express means genes produces certain proteins, or they produce, give instructions to do, they are like a computer code. They give instruction to do certain things. So certain thing doesn't happen accordingly if there is a faulty gene. So mutation, that is known as mutation. So mutation occurs, so that would give a faulty expression. Say that one eye will be like this and one eye will be like this. So these kind of things happen. So these are genetic things. So medicine also, so you can have the genetic testing. It involves the direct examination of DNA molecule for the mutated sequences. You get the DNA from your body, test it for whether the sequence is there or not, some mutation is there. So I can determine this is the problem. Your child, suppose if I have this test, I can determine whether my child will have in future this disease or not. So that way this testing can help. These are the application in medicine I am talking about. Suitability, through biotechnology, you are trying to determine the body's suitability for the particular medicine. Then you are using genetic testing to determine whether this genetic diseases are present in your progeny, will be present in your progeny or not.
So these are the, this is too much, I would say too much of English. They have written too much hi-fi, but they won't be of much use. But still you have a knowledge, because you have to write, have a general knowledge about a lot of things. Have a knowledge just in case you add something little bit of this as well if I write. Then RDT I talked about, a recombinant DNA technique. The RDT processes had a great impact on the area of healthcare by enabling mass production of safe and more effective therapeutic drugs without inducing unwanted immunological responses. Example of applications include gene therapy, molecular diagnosis, etc. Same thing. Gene therapy means you are using medicines which modify the genes. So your genes are try to be corrected so that you get the you don't have the problem diseases and all those things. So these are the things. Application in medicine is pharmacogenomics, pharmaceutical products, genetic testing, RDT, recombinant DNA technology. I told you, gene editing. You take the cell gene, recombinant, you put some other gene, the desirable gene. So you make that gene, desirable gene in that sequence, and you have that expression, that correct thing is, you get the correct. Like I put, say I want blue eyes for my children. I tell this technician, you put do the modification of my gene, this thing, you put gene for blue eyes in my children, so that when that thing happens, so the thing, baby will have the blue eyes. So that is this is what gene editing is. This is very powerful technology. I say you can have. I told you about the white race theory of these Germans, na? Did I tell you? This is what they had, this theory that they would have the superior children. Gene Aryan theory. That they at that time these things were just emerging. Gene editing was not there, but they had about the theory of genes. They had some knowledge. So they had this theory that we are a superior race, and we will continue to have a superior race people, and we would have children or the next generation who are white, fair, white, blue-eyed, blonde hair, those kind of race. And we won't be having people with disabilities, these legs like this, or blind or deaf and dumb. So they wanted to edit the population. You get me? So this was what thinking was, but that is against the nature. You get me? We have to. Science is not so advanced. Maybe in future, in 500 or 1000 years, some advancement will be there in science. But at the time, those people thought we would have a better race like this, so we'll edit the population and have healthy and better people of these traits, blue eyes, white. So this is possible. With biotechnology, this gene editing, this has become possible. You can edit the genes, but this is not a practical technology. You cannot go to this thing, biotechnology center, say I want my designer baby like this. It is not possible. Second, there will be a law. Many countries there are law. They won't. Somebody who has also, you can say, let me have a baby who's a genius, superior. That is possible. You modify certain genes, but like you said, there are chemicals. You don't know what are the side effects would be. There might be some other side. You might get the blue eyes, but you might get, blue ear as well. So it might be possible. Because science is not that perfect, that genetic side effects might happen, still happen. Otherwise, this technology would become very commonplace. Everybody would be using this, but it's not happening. It is not happening. So this means what? It is still in very experimental stage. These things are happening. This technology is there, but these are very experimental, very limited level. This is not at the scale. Like in the case of farmers, if we had the best plants, why we are still having problem with agriculture? Why the food gets spoiled? Why so much food is wasted? Why droughts, when there are droughts or there are hailstorms of the food, these things are destroyed. Why? because still this application is very limited. These things are applications are there, but very limited as per what I think. So they may become mass oriented in future in maybe 500 years and 1000 years. Like earlier you used to be this bullock carts. Now the vehicles, everybody is using vehicle. It had taken about 150 years for this thing to become generally common, mass oriented. So this maybe in 115 years biotechnology, these things might become with better regulation, with the regulators ensuring that unethical hacking and these things might not happen. They had better control. So I'll try to tell you also later on. There is a concept called DNA fingerprinting. Everybody has a unique DNA. Like my fingerprint has my DNA fingerprint is unique. Sequence of DNA bases and these are unique. Now what happened? Like police wanted in one case. One police wanted they said terrorist or I don't know what, they wanted that person to be identified. Like in case of Osama bin Laden, what happened? How they identified him? He had changed his appearance now almost. How they identified? Through the DNA. They took his DNA, they had the earlier DNA with them. So they matched. That is not a legal thing. Legally you have to go to a magistrate, get permission, then do it. But still they are doing it because they wanted immediately. They took this thing sample. They involved the person in the accident, collected his blood sample and matched the DNA, identified the person. So these things legally, otherwise you have to go to magistrate, get the permission, then approach, seek consent, but those things practically don't happen. Things are very, very different what practically happens and what is in the book, statute books. Anyway, then application in agriculture. I talked about the medicine. I gave a flying view of the medicine. Now the application in agriculture. So like how does it affect biotechnology affects the agriculture? Crop yield. One or two genes may be transferred to highly developed crop variety to impart a new character that would increase its yield. That is, you take a crop which is good. Generally, what hybridization is, grafting you have seen. What happens in grafting? That is natural biotechnology. You take from one plant which is more productive to the another plant, then you cut and put it splint there, then tie it. That is natural. Same species you are putting there. Like in case of kinnu, sweet lime and orange. You are grafted from one plant to the, say you have taken this thing branch from a sweet lime and put it on the this thing orange, grafted it, so you get the kinnu. So same thing that is happening naturally. They were doing this, but with biotechnology, what you are doing? You are going to the molecular level, cell level. You are changing the genes. You are finding out which genes. Generally, the expression when you are doing it in crude manner, you are doing it on a scale. But when you are going deep and you know the precise tools, this is the thing, then you are more effective. You are using less quantity. Like I talk about the bio fertilizers. What bio fertilizers are? Like chemical fertilizer using NPK, this potash, sodium, nitrogen, all those things. So, but with this, with biotechnology, you're using the particular bacteria which fixes nitrogen. So you're putting them directly in the root, not putting lot of bags of urea or all those things. So you're putting a small quantity of in the roots of the plant. So you're getting the same result. So you can increase the yield of the crops, make them resistance to diseases and all those things. That I'll come later. Okay. Then reduce vulnerability of crops to environmental stresses. This is another aspect. Increase the yield, then to reduce vulnerability of crops to environmental stresses. Stresses mean what? Heat, rain, this hail and all those things. Crops containing genes that will enable them to withstand biotic and abiotic stresses may be developed. Biotechnologists are studying plants that can cope with extreme conditions like drought. So biotechnologists working on this, that what happens like when suppose there is a lot of sunshine, you have a plant, it will wilt. Why? Because maybe the moisture or due to, but you make it sturdy. You put through gene, you say you put a gene of cactus into a rice plant. Cactus can survive in extreme condition. So that will trait it will acquire a rice plant. So it can survive the extreme condition. So this is what biotechnology is. So one thing, this thing, reduce vulnerability of crops to environmental stresses. Then increase nutritional qualities. Increase nutrition qualities means you have protein, you increase the amount of protein in this thing. You don't get the protein supplement, you increase the protein amount in the particular fruit or something. So this thing. Increase nutritional qualities. Protein foods may be modified to increase their nutritional qualities. Then improve taste, texture or appearance of food. So modern biotechnology can be used to slow down the process of spoilage so that fruit can ripen longer or the plant can on the plant and then can be transported to the consumer with a still reasonable shelf life. That is, you are having increasing the ripening stage of the fruit. Generally, what happens? Fruit would ripen sometimes very early, sometimes very late. So you are making it you are changing the way the fruit's qualities, traits, that it slowly it ripens, so that it doesn't get spoiled. And similarly, like then this insects would come and when the fruit is ripening, the insects would come or the birds would come. So you are also giving that qualities that those things don't attack the fruit. So you are preserving the fruit. So spoilage you are reducing. Then E point, reduce dependence on fertilizers, pesticides and other agrochemicals. For example, introduction of herbicide tolerant crops has the potential of reducing the number of active herbicide ingredients used for weed management. Same thing, weed management, like if the plant doesn't, you changing some trait, so when you are using the herbicide, the plant doesn't get killed. Like you trying to destroy the weeds get destroyed, but the plant has the capability to withstand the herbicide effect. So these are the things. Production of novel substances. For example, oil seeds can be modified to produce fatty acids for detergents, substitute fuels and petrochemicals. Potato, tomatoes, rice, tobacco, lettuce, safflowers, and other plants have been generally engineered to produce insulin and certain vaccines. So these, like you are modifying, giving some properties, so you get the certain outfit. So application in this health and in this agriculture I have talked about. Now you ask me questions. I have talked a lot. Tell me. Ask me question. Yes. Do ask anything which strikes your mind. Make your mind fertile. Like biotechnology, use biotechnology on your mind. That you are making it fertile. So many ideas, full of ideas, bubbling with ideas. Have you understood what I told? Then tell me what I repeat, what I have told. Tell me what I have told. In medicine and agriculture. Biotechnology application. Medicine or agriculture, both. Okay, good. Shelf life, okay. That is a recent one, which is recency, memory. This is known as memory recency. You remember what is being told latest. Then I talked about plants, I talked about the disease control, biotic and abiotic pressures. So you have forgot only the last and first part. Okay, then health, medicine. Pharmacogenomics.
Make more body resistance to any drugs.
To suitable drug as per the body's response. Okay.
Recombinant DNA technology.
Okay. What is that? RDT?
Sir, it means recombinant means making our DNA.
Not our, any things, organisms.
DNA capable
Okay, forget it. That way it will take a lot of time. Okay, then I'll come to bioremediation. This is another aspect of biotechnology application. What is bioremediation? It is the use of organisms, usually microorganisms, to break down pollutants in soil. Write down, this is important. Bioremediation, BIO dash REMEDIATION. They might ask you this question sometime in future. Bioremediation. What is this? It is the use of organisms, usually microorganisms, to break down pollutants in soil, air, or groundwater. This is like bacteria and all those things. I told you now about oil. Sometimes oil spills are there. So it is the use of organisms, usually microorganisms, to break down pollutants in soil, air, or groundwater. It can be classified as in situ or ex situ. In situ bioremediation was treating the contaminant at the site, while ex situ involves don't too much. Some of bioremediation techniques are phytoremediation, bioleaching, land farming, bioreactor composting, biomedication. This is too much. No need. Then I come to the animal biotechnology application I am talking about. Agriculture, I talked about medicine, then bioremediation, that is environmental. Also this, you talked about the codes, na? This environmental application in environment, treating of pollutants. So pollutants like in oil spills and all those things. This green, but not green, environmental biotechnology. Branches I told you, na? So this is application in that field. So you link that way. Branches link how the application, how it is being applied. Then animal biotechnology. In animals, biotechnological techniques are being used to improve genetics and for pharmaceutical and industrial applications. Animal cloning allows for replication of selected animals. Genetic engineering using RDT alters the genetic makeup of animal for selective purposes, including producing therapeutic proteins in cows and goats. This is animal husbandry, I told you, application. You're changing the genes and all those things using RDT, and then you're getting good milk, wool, and all those things. Meat, good quality meat. That is through genetic cloning. You have a good quality. Earlier used to be like the horses they would breed, na? For good quality horses. No, through genetic manipulation you have the good quality this thing animal. Then you clone it. Clone means copy that DNA and put it into another animal and this thing. So you have this cloning. Okay, then industrial biotechnological. This is the fifth one. It is used to improve efficiency and reduce the environmental impacts of industrial processes like textile, paper and pulp, and chemical manufacturing. For example, biocatalysts such as enzymes are used to synthesize chemicals. Using biotechnology, desired enzyme can be produced in commercial quantities. Using biocatalysts, the chemicals can be produced more economically and more environmental friendly. And an example would be substitution of protease in detergent for other cleaning compound. So that is, you are using these biotechnical products like enzymes and all those things for producing industrial products like enzymes for detergent for detergents. Utensils you using the detergents for clothes. So those things you can be produced through the biotechnology method. Biotechnology method means you have a bacteria which is producing that kind of enzyme. So this is very fast and dependable, very fast. Otherwise, chemical process, you have the big plants, then you have this baking soda, this thing, and those things take a lot of time and effort. So these are very quick processes based on biotechnologies are very quick and give good results immediately. Any questions? This is something I've covered in a hush very fast manner. Tell me. Any questions? Whatever. Because I talked a lot about this thing, biotechnology, in this video lectures. That you have missed all those things. I covered a lot of things. So this is just a snapshot of whatever is there. This is as per your notes. Still, I will try to come again on those topics. Tell me anything. Ask. Good questions you are asking. Bioremediation, those things. Where is the printing press in India? Currency printing press. ISP Nasik. Nasik. It is. Here it is. Nasik. Where you print currency notes. Char bits. Maharashtra Nasik. ISP Nasik. So these are the tidbits. Okay. So tell me, anything you ask not related to biotechnology or science and technology, other things about civil services, ask. Anything to do with your preparation. Today notification would come, na, I think, after 2 o'clock. I think after that you have to fill the form. 24th till 24th is there. So you are not appearing for this year, na? Not. You are appearing? You are also not appearing, na? Next year you are. Good, good. You are appearing for this year. Good. Yesterday, day before yesterday, I was talking to somebody. He said this guy had appeared twice for the civil service exam. He said, "Oh, I have got 4 more attempts." I was just thinking, 2 you have wasted, and you're thinking you have 4 more attempts. You'll be wasting 4 more years of your life. So you should be very confident that you should clear in the first attempt, at the most second attempt. Not waiting for 5, 6 attempts, writing, wasting time. You should be very confident. You should prepare like that, like a professional. That should be your goal. Otherwise, you'll be writing, then gaining experience, then writing, then again gaining experience. That would be a total wastage of time of your life. Then when you look back those years, then you would see what you had wasted in your life. Anyway. Okay, now I'll come to this thing. You are talking about the regulation. This is as per your notes only. Nothing more, nothing I added. So this regulation, this is biotechnology related policy developments in India. So first was, these are all bills, nothing has become an act. These are all bills. This is there in your notes. The first one is the DNA technology use and application regulation bill. And this is the DNA technology use and application regulation bill. So in 2019, Cabinet cleared this bill to regulate use of DNA technology in India, 2019. There are a lot of things that you would find. These are 2015 policy statements, vision statements, and nothing had happened. Nothing had happened. But still, these things are there because you'll get an idea of what is there in the notes. So, DNA technology use and application regulation bill. It is a bill, not an act. So, bill. You know what bill is? What is a bill? Bill is just a formal, this thing is you have introduced into the parliament, or you are planning to introduce into the parliament for approval to make it to an act. What is the difference between a bill and an act? Okay. That becomes an act. What is a bill? Bill means it is not come into force. Till the time it is not into force. Something you have a bill, so many bills. Every day there is a bill in private members' bill. Even an MP can say, this is a bill. Then there is a voting. Sometimes there is voting, verbal voting. Then sometimes there are votes are cast. So different procedures for enactment of bill. So this is part of your Indian polity. So anyway, I'll come to this DNA technology use and application regulation bill. It is still a bill. So in 2019, Cabinet cleared this bill to regulate use of DNA technology in India. This is 2022 and nothing has happened. It allows the profiling of victims, those accused of crimes, and those reported missing, and storing of their DNA into national and regional data banks. It also aims to set up a DNA regulatory board. This is what you were talking about, na? that regulation. It is not there, still not, no fine print of this thing has come out. So they have talked about this DNA regulation technology, use and regulation technology. So this bill is there. I don't think this is of much use, but still they have filled 2-3 pages of your notes with this. I'll elaborate on this thing. So need for the bill. It seeks to create a regulatory framework for obtaining, storing and testing of DNA samples of human beings, mainly for criminal investigations and to establish the identity of a person. This answers your questions. So this is what the aim was, to have a data bank, regional data bank as well as the national, where you store the information. They are very cleverly they have put this thing that only with the permission of the people the information would be stored. And the criminals, for the criminals you don't need, I think some kind of they have put some breaks in this for getting the information of criminals as well, but generally for serious crimes they have foregone with the permission of the accused for storing of the information. In case of certain people, missing people, suppose like you have seen in the movies, na Kumbh Mele, those people are separated. So how do you unite those people? So you have this, if you have the DNA of the two people stored, you can match your parents or somebody, so you can match with the missing person's DNA to say that yes, you can identify that. So these are the well-intentioned aims of this bill. So this is the need for a bill. Second point, it says DNA analysis offers substantial information which, if misused or used improperly, may cause serious harm to individuals and society as a whole. So that's what wise thinking is, that they think that DNA analysis can be misused. So we should have a regulation oversight over the working of this sector. Regulation, use and application, DNA technology. Then the third is proposed law seeks to bring in a supervisory structure to oversee these practices and frame guidelines and rules so that DNA technology is not misused. To this end, bill proposed to set up two institutional structures, a DNA regulatory board and a DNA data bank at the national level. So key provisions of the bill. This is there in your notes. DNA regulatory board. It would frame rules and guidelines for DNA collection, testing and storage. It would be the final authority to authorize the creation of state-level DNA data banks, to regulate the quality of DNA labs by providing for mandatory accreditation and regulation of DNA labs. Testing of DNA samples can only be done at labs that are authorized to do so by the board. So this is what regulation is. You have these labs, diagnostic labs. So how do they work? Generally, you have, which lab you have used sometime? Which diagnostic lab you use? Or somebody in your family, have somebody used this labs? Tests, you have not done tests recently for anything? Or in the past? Which one? Which lab? You don't know. You? Which one? Lab path lab, okay. You do. Lab, which name? Vimto, Stimto, Sigma. So these are what happens, these labs, they can also misuse your data. Like when they collect this blood, they can, if they are not regulated, they can use this blood for a lot of different purposes. They can send it for DNA profiling. They can, without your permission, they are taking the blood, they take the blood, they can use it for DNA profiling as well. You don't never know. Then they can blackmail you. You are not the son of your father. They can tell you something like that. They can do, these are the things I am telling. But they won't be doing this thing. Why? Because they are regulated labs. If something happens, there will be a criminal complaint against them. So these labs are regulated. But DNA labs, DNA use and application technology labs, they are not regulated right now. There is a board, NABL, National Authorized Board of Laboratories, something that controls these laboratories, these microbiology laboratories. But for DNA testing and all those things, there is no board. So this bill is proposing to set up board at the center and the regional data banks that for the purpose. So this is what it is. Regulation, you understand what regulation is? RBI, what is RBI? What? It is a regulator. It regulates what? Other banks. Other banks means the banks. What banks can do a lot of frauds. They can give money. Say they can say I'll charge 50% for any loan. I'll give you 0.5% for your deposits. But it doesn't happen. Why? Because RBI is regulating. It's saying there is a range. You give 3% for saving bank account deposits, 5% for this thing. It gives a range. 3 to 5% or 3 to 7% according to them, and you charge loan as per the market condition, maybe 7% or 8%, not 50%. So you don't go for the predatory practices. So they are regulated by somebody is watching their working. It's working. So oversight is there. So this is what this bill proposes to do, but nothing has happened. So for so long things have not moved. Just imagine the way the things are working. So this bill is there. So regulate the DNA use and application technology, DNA technology. So this thing, like still it is a gray area. Anybody can go and get your this thing and misuse it for other purposes. So this is what it is. So DNA Regulatory Board. It would frame the rules and guidelines for DNA collection, testing and storage. It would be the final authority to authorize the creation of state-level DNA data banks, to regulate the quality of DNA labs by providing for mandatory accreditation and regulation of DNA labs. Testing of DNA samples can only be done at the labs that are authorized to do so by the board. Then DNA data banks. Bill provides for establishment of national and regional data banks. There would be repository of all DNA samples collected from various people under specified rules. The national regional data banks are required to store the info under one of the 5 indices: a crime scene index, a suspect under trial index, and offenders index, and missing person index, and unknown deceased person index. So this is lot of information is there. You get some idea. If you read that notes, if you find something difficult, do ask me. Then the third point is, although information for DNA can yield much into about the person, the databases are supposed to store only those information that is necessary to establish the identity of a person. So these are the things structure they are trying to create. Have a national data bank at the National Regulatory Board at the top, and then the regional data banks where these things samples would be stored and analyzed, and the data would be stored. So I'm not going into much of this, but if you have any difficulties understanding this thing, regulation, you understand regulation. So still I'll try to complete. Permission for DNA profiling. While preparing a DNA profile, bodily substances of person may be collected by the investigating authorities. Authorities are required to obtain consent for collection in certain situations. For arrested person, authorities are required to obtain consent. If the offence carries a punishment of 7 years, if the offence carries more than 7 years of imprisonment or death, consent is not required. What is Indian Penal Code? Indian Penal Code. What is that? It is a code of penalties and all those things. If somebody commits some crime or all those things, so Indian Penal Code has defined processes of how to go. This is for criminal processes. Indian Penal. CRPC, Criminal Procedure Code, similar Criminal Procedure Code. So they prescribes the various procedures how the criminal proceedings have to take place. So and the punishments, Indian Penal Code punishments, the different punishments. So these are the legal framework. For certain crimes, there are different punishments. You have to pay the fine. For certain crimes, you have to serve the sentence. You have to go to jail. So for some crimes, it is up to 3 months, 6 months, or maybe 1 year. For some 7 years. For some more than 7 years. For some life imprisonment, and for some even death. That is capital punishment. So these are the, depending on this is what they classified, depending on this thing, depending on the severity of the crime, you have to be your permission, assent, or you're giving you're giving assent or not giving assent is required. This is the 7-year offense and all those things. This is all there in your notes. If the person is a victim or a relative of a missing person or a minor or disabled person, the authority is required to obtain the consent of such victim or relative or parent or the guardian of the minor or disabled person. If the consent is not given in either case, the authorities can approach the magistrate. People whose DNA samples have been collected either from the crime scene or through voluntary written consent can also request the removal of their info from the index. Like you have in the Google, na, your information is there. All the information is there. So can you remove that information? You can write to Google that you kindly remove this. They would ask for certain clarifications and then they can remove. Because a lot of people got into trouble for having their information on Google. So somebody was involved in some petty crime, so they couldn't get recruited by somebody because they found out that they were involved in some petty crime. So this information, same thing that the person, accused person or a concerned person can get his information removed from. This is what bill says. This is a lot of good intentions, but not come into force. Then use of DNA data. Under the bill, DNA testing is allowed only in respect of matters related to the schedule to the bill, such as offences under in IPC 1860 for paternity suit or to identify abandoned children. So what for you using the DNA data? Why are you going for a DNA test? That is paternity suit. You don't understand what paternity suit is? To determine whether the child is legitimate child of the concerned parents or not. So that is a paternity suit. So this is again all those things. Or the abandoned children. Somebody has abandoned his child, his or her child. So you can identify through that whether the person that the abandoned child belongs to the particular person or not. So these are what intentions of the. Then the offences. The bill specifies penalties for various offences, including disclosure of DNA information or using DNA samples without authorization. For instance, disclosure of DNA information will be punishable with the imprisonment of up to 3 years and a fine up to ₹1 lakh. So these are the things. So the bill. What do you think? Government should have acted fast and got it enacted or not? Because they've got so many bills enacted. Reorganization of JNK in 2019. But why then don't? This is a sort of the thing. Why don't they are not concerned with these such things like biotechnological application, misuse, or all those things? They do vote banks. That is correct. That is correct. So they are not. But they say they are bothered about the welfare of the people. So this is not that is welfare of themselves. Na? They are bothered about the welfare of themselves, not the people. Why these things are taking so much time? You cleared, the cabinet has cleared the bill in 2019 and this is 2022. Still it's implemented. And then also your notes you will find there are policy statements from 2015 to 2020. Lot of high sounding words like how to implement this biotechnology. Any which way, no budget has come. You have yesterday the budget speech was there, so you missed this. What do you find? How do you find the budget? Tell me. So this is all in the realm of speculation and possibilities only. Nothing is happening on the ground. So that's why we as human beings are not advancing. Biotechnology is there, such a beautiful technique, but we are not we still have diseases, we still have problems. Why? Why? Because of the human nature. We are fighting and we are living lives on different considerations. Any which way. So tell me, any questions on these bills or on the applications part I told? This definition which I told you, this is very important. You do write this definition again because this definition is not in your notes. I told about, this definition of which, where is that? That is very good definition of biotechnology. This is the definition you should know. The developments in genetic engineering, cell culture, immunology, protein. I think you have written. Just check. The developments in genetic engineering, cell culture, immunology, protein engineering, fermentation technology, collectively called biotech. Collectively, these are the things. Otherwise, your notes refers to the application of various biological living organisms and processes for production of useful substances or effects in medicine agriculture. So this is generally, he has given just techniques and tools, but this definition, the earlier first definition, it covers everything what biotechnology is. Immunology, protein engineering, genetic engineering, fermentation technology. These are what applications are. In immunology means in health, and then genetic engineering in industry, in animals and aquaculture and all those things. Fermentation technology, food, protein engineering. This is again health and medicine. So these are some things. Ask any questions you have. Anything you ask about your preparation, if you have any doubts, anything. I think you are quite confident that way, prepared that, but still you never know. You, like the essay questions, yeah, I think you are stumped literally. So they have asked, because generally when they ask these questions, they have been asking these questions, but in the optional papers, in the form of this, there used to be the questions. Earlier there used to be questions, they would ask write in 100 words, then they would give a quotation like this. This time they had replicated in the essay paper. So they would ask you to write now in 1000 words. So in the optionals they would give you some quotation and then ask you to write on those in 100 and 150 words, small paragraph. I don't know still they're doing it or not. First question or second question would be like that. So tell me any questions or anything you want to ask. Think.
DNA जो होता है वो पेरेंट्स और चाइल्ड के सेम होता है ना, 100% मैच करता है.
It should match
100%.
100%.
इसमें मेरे ब्रदर का मेरे पेरेंट्स हमारे सेम DNA.
Yes.
सर DNA फिंगरप्रिंटिंग जो होती है फिंगरप्रिंटिंग, सर DNA फिंगरप्रिंटिंग.
DNA fingerprinting means like this fingerprints. Now you have got certain marks on your finger. So these are unique. Everybody has unique pattern. You have got different pattern. I have got different pattern. So DNA fingerprinting is what? My DNA is what? DNA is basically bases A, C, T, G, like this. They are arranged A, C, T, G, T, C, A, G, like this. If there is, this is unique. For me, it will be A, C, T, G, C, A. For you, it will be A, T, G, C, like this will be the sequence. Everybody has got different sequence. So those sequences, that is the uniqueness. That like fingerprint is unique. So DNA fingerprinting means unique sequence of your this base pairs in your genes. I'll explain it to you.
इससे ये भी हो सकता है अगर मैं कुछ मतलब मर्डर वगैरह कर दूँ, ठीक है, मे भी मेरे DNA उनको मिल जाए, मे भी बात हेयर के थ्रू या कुछ, तो उसमें भी मेरी फैमिली भी तो जा सकती है ना. हमारा DNA सेम ही होगा, उसमें वो पता कैसे लगेगा?
DNA unique. DNA doesn't mean your father's. That is what biological tracing is. That's how they have traced so many criminals. They identified the DNA belonged to a particular family. Then they could identify the criminal. But DNA doesn't mean your father's DNA will be like A, C, T, G. There will be some differences, uniquenesses in there. Identically, it not will like 100% identically A, T, C, G will be same. Half of your genes have come from your mother and half from your father. So your father and your matching will not be same. Like your father had A, T, C, G, you will be have A, T, C, G. You maybe have A, G, T, C, something like this. You are not 100% your father and 100% your mother. Half of the genes have come from your mother and half from your father. So this is a good question you ask, that anybody, if you have committed a murder, anybody from your murder. But fingerprinting is unique. Every DNA is unique in that sense. Not it is, here is 100% of your father. 100% in the sense, 50% of your father and 50% of your mother. 100% that way it is 100% of your parent. So some criminals, they nabbed like this, na, through nowadays they have this websites which they tell the history, your family history. They can tell you where from you have come, from, say 500 years back. So these are known as what is that? They trace your ancestry, like where from you have come. So using this tool, this biotechnological tool, the police in America, they identify a criminal who had committed crime over 50 years back. Because of this genealogy, they could identify a group of certain particular families where this person belonged through database. That is again ethical concept which comes whether you can use the data without their permission or not. But criminality was involved, so they used this data to identify. After 50 years, they could identify. He left no clue or no any kind of sign of his having being involved in the murder. He was a police cop. But they could identify through this technique after 50 years. Genealogical sites, they could trace out this ancestry through a DNA, that this person belongs to particular ancestry, and then they zeroed in on the particular individual. So that is a DNA fingerprinting. Science and technology? I'll take up this IT, computers, robotics, lasers, and what more is there than this? I think that is sufficient. This type of question they would ask you from biotechnology. There's a lot of things that agriculture is also part of science and technology. Environment is also part of science and technology, but I won't be taking it. I will go as per your notes only. Then we have covered space, defense, and nuclear technology. So this is, I think, more than sufficient. What about other portions, your ethics and you're not started? Or should have started by now?
Ethics और international internal security वो रहेगी
That's you have finished. Polity as you have finished.